It is a simple treatment that can be applied to any stem cell line or differentiation protocol to prime cells for differentiation. All one has to do is to add DMSO at 1 to 2% into the cell culture maintenance medium.
The main advantage of this technique is that it allows one to significantly increase the differentiation potential of pluripotent stem cells into any lineage of choice by priming cells for differentiation.
DMSO treatment increases the chance that cells will differentiate from patient-derived iPSCs, allowing them to be used for cell replacement therapy, disease modeling, and drug screening. It can give rise into the early onset of regulation that controls cell proliferation, differentiation, and specification during embryonic development.
We have demonstrated that the DMSO treatment improves differentiation in nearly 50 human embryonic and induced pluripotent stem cell lines, while other laboratories have shown that it's beneficial for stem cells of other species such as mouse and primate.
1 day before the onset of differentiation, add DMSO at a concentration of 1 or 2% for 24 hours into your typical maintenance median. 24 hours after the DMSO treatment, replace median and proceed with your usual differentiation protocol.
Grow cells according to manuscript directions. When the cells reach an appropriate confluency, dissociate them and prepare a single-cell suspension. Count the live cells with a hemocytometer or automatic cell counter using a viability marker such as trypan blue. In order to achieve 80 to 90% confluency within the 24-hour DMSO pretreatment, plate the cells onto a coated 6-well plate at a density of 500,000 to 1 million cells per well. Incubate the cells for 24 hours at 37 degrees Celsius.